This video has been funded through an educational grant by Biocon Biologics Ltd. Biocon Biologics Ltd has had no input towards the content of the interview or the video. The 2024 ESMO Congress, held in Barcelona, Spain from September 13th to 17th, 2024, featured 269 sessions and 2,186 abstracts. These presentations covered the latest in translational cancer science, practice-changing guidelines, and interdisciplinary approaches to patient care. Professor Ahmad Awada spoke to us following the conference to summarize the latest in breast cancer research and where these developments may lead for future treatments.
We are living a really nice evolution, even to say revolution, with the development of many interesting agents in solid tumor in general, as well in breast cancer. I think the dream of the oncologist. I am in this field now since more than 30 years. I remember one of our dream as oncologists is to have targeted chemotherapy. I think we reached this objective to some extent today with the development of ADCs, which could be considered targeted chemotherapy. It's clear we have a good benefit in many solid tumors, including breast cancer, but also in many other tumor types, bladder cancer, ovarian cancer. With these ADCs, we can say we improve significantly the efficacy of chemotherapeutic agent, but we cannot say we decrease the toxicity. We can say we change the pattern of toxicities with the ADCs compared to standard chemotherapy. Of course, ADCs are one part of this innovation. The second, tyrosine kinase inhibitor and other small molecules. In checkpoint inhibitor, I hope that we have now bispecific, even three specific drugs, which in part target the cancer cells and another part target the immune cells. I am optimistic about the development of tumor vaccine now, because the tumor vaccine with this vaccine, which target sometimes dozen and even hundred of neo antigens. And so this is the reason my optimism about vaccine in some situation. We started to see interesting activity in melanoma, metastatic melanoma, as well, even in non-small cell lung cancer. I think radioligand therapy is a really good field too. We know radioligand is efficacious in thyroid cancer, in neuroendocrine tumor, as well in PSMA positive prostate cancer. But I'm expecting that radioligand therapy will emerge in other solid tumor with other target. CAR T possibly in niches, and that's the most important development. I am happy. I think we can say one thing's important. With all this development, it's really important to innovate in the clinical research methodology. That if all this development innovation, it's important that the patient will have access to this innovation, because if there are no access to patient, this innovation remains invention and not innovation.
What data was presented from the Icarus Breast 1 study?
This study is phase 2 trials, around 100 patients, using as a new agent, an ADC, patritumab deruxtecan, targeting HER3. As you know, HER3 is a poor prognostic factor, is responsible of resistance to PIK3CA inhibitors, as well to endocrine therapy. And so what is interesting in this trial, even if this trial is phase 2 single-arm trial, they gave the drug, the ADC, to patient 100% pre-treated with CDK4/6 inhibitors. 35% of the patient already received PIK3CA inhibitors. And interestingly, too, that this this trial included a patient with HER2-0, around 40% of the patient, 1 and 2, and not HER3. More importantly, in this trial, there are a major translational component which gave some interesting information which need to be validated in large scale. And the main objective of this trial, overall response rate, and interestingly enough, in patients already exposed to CDK4/6 inhibitor, as well in some extent to PTCA inhibitors, the objective response rate was around 53.5%, 2% complete remission, and 51.5% partial response. In terms of tolerance, overall was okay, as expected, fatigue, GI toxicity, some alopecia. And I would like to comment a little bit on the translational component, because that's really a huge component. I think the main message that HER3 expression didn't correlate with the responses. That's important. It seems that HER3, the distribution of HER3 DXd is important in the whole population and in patients responding to patritumab deruxtecan. And finally, there are upregulation of gene involved in immune response in the whole population, as well in patients responding to this therapy. So this put this ADC, patritumab deruxtecan, as a really important drug to be developed further in hormone receptor positive, HER2 negative metastatic breast cancer, and why not later on in the early setting.
What was the latest update from the Destiny trial series?
I think the Destiny Breast specifically presented at ESMO was Destiny Breast 12, which include the patient with brain met and patient without brain met. This is a phase 3b/4 study. So this study, Destiny Breast 12, included patient. Let's focus on patient with baseline brain metastasis, around 263 patients. 106 were active brain met, that's mean untreated and active, but not, there are no need to immediate local therapy, or pre-treated but progressed. So this is around what's called active brain met, and there are another group with 157 patients with stable brain met. In this trials, which include patients already 100% pre-treated with trastuzumab, 87% received pertuzumab, 40% received TDM1, and 60% received tyrosine kinase inhibitors. And importantly, to mention too, that visceral metastasis in this group of patients was around 25% in the lung or in the liver. And so we have the 12 months PFS for this population with brain met, with around 62% for stable, and for active brain met, the 12 months PFS exactly the same percentage, 62%. The median PFS was around 17 months, which is really good. The CNS objective response rate was 72%, mainly in stable disease, 79%, and 62%, which is not bad at all, in active brain met. And the overall response rate in patients without brain met was around 63%. So there are no difference, basically, for the objective response rate between patient, objective response rate in brain met compared to other side of the body. The 12 months overall survival in patients with and without brain met was around 90%, which is outstanding and interesting. And finally, pneumonitis, still a major issue in patients with brain met, and there are some patients, 2.3% of grade 5, that means death due to pneumonitis, but some of these patients with co-infection, probably because this patient received steroids in the same time for brain met. For the rest of these side effects are exactly what we know from trastuzumab deruxtecan. I mean by that GI toxicity, particularly, and fatigue, and so on. So the conclusion of this important trial that this drug now is really active in patients with and without brain met, particularly in patients with a brain met, and this drug is added now to the therapeutic algorithm for the treatment of patient HER2 amplified disease with brain metastasis. And now we have two approaches for brain met in HER2 amplified tumor. We have the tucatinib-based therapy. We have the trastuzumab deruxtecan. We cannot compare the efficacy because there are no head-to-head comparison, but we can know exactly the side effect of one agent as well, the tucatinib-based combination. And so we could now, let's say, tailor the treatment, taking into consideration at least the expected side effect. So we are advancing really well in the management of active brain met as well as stable brain met in patients with HER2 amplified metastatic breast cancer.
While those trials were successful, what about the Capitello 290 results?
Yeah, Capitello presentation is interesting from the methodological point of view, but unfortunately, this trial was negative. So the rationale to develop this trial with capivasertib, which is PIK3CA, AKT, P10 inhibitors, is because we have two studies which shows in phase 2 studies which showed interesting data, the PACT trial and Lotus trial, which, let's say, push to do phase 3 trial in triple-negative breast cancer, first-line metastatic disease, comparing capivasertib Taxol to Taxol plus placebo. And around 400 patients entered in each arm. 30% of the patient had PIK3CA AKT P10 gene alteration. And the side effect, as expected, GI toxicity, hyperglycemia, and rash. The primary endpoint was overall survival, and functionally no statistically different difference in survival between the two arms. Also, for the objective response rate and PFS favor capivasertib in combination with paclitaxel. So in summary, this study didn't met the primary endpoint, which was overall survival in the whole population, as well in PIK3CA AKT P10 alteration. So for the moment, this drug is active mainly and beneficial to patients with hormone receptor positive, HER2 negative, in combination with fulvestrant in the metastatic setting after exposure to CDK4/6 inhibitor, and in patients with PIK3CA AKT P10 alteration.
Finally, is immunotherapy making any breakthroughs in treating breast cancers?
I think there are an update and on the overall survival data of Keynote 522 study of pembrolizumab in combination with chemotherapy in the early setting of triple-negative breast cancer. We know that pembrolizumab in combination with the best chemotherapy we have in our hand today, anthracycline, cyclophosphamide, carboplatin, taxane, in combination with pembrolizumab, in terms of PCR rate, is around 2/3 of the patient. We know that invasive disease-free survival is improved significantly in the pembrolizumab chemotherapy arm. And we have had at ESMO, last ESMO, an update on survival. And the good surprise that there are 5% overall survival benefit. The delta is 5% in favor of pembrolizumab in combination with chemotherapy. But I think it's important to mention too that the patient who benefit most in terms of survival, patient who did not develop PCR after chemo pembrolizumab therapy, there we have a major, let's say, advantage in terms of overall survival as well for invasive disease-free survival. So in summary today, in patients with early breast cancer, triple-negative breast cancer at high risk should benefit now in clinical practice from the combination of chemotherapy and pembrolizumab. In patients with residual disease, this patient should continue pembrolizumab. We could discuss case by case if we will add capivasertib. For patients with complete remission, I think the question is still open. Should we pursue with pembrolizumab in the adjuvant setting in patients with PCR after chemo and pebro in the neoadjuvant setting? This is an open question, knowing that two studies in the adjuvant setting with immune checkpoint inhibitor without immunotherapy in the neoadjuvant setting showed negative result. I'm thinking about Alexandra trial, for example, was adjuvant trial with immunotherapy was negative.